Hello everyone, welcome back to Food Feeds. In this video, we are going to learn about carbohydrates. As you can see, this is just a part 1. I have divided this topic into number of parts, which is a small parts, so that it's easier for you to learn by watching the short videos rather than going for a longer lectures. And it would be great if you have a pen and paper with you so that you can jot down the notes as the video progresses, so that by the end of 30 minutes, you will have the notes ready for you for this particular topic and you'll understand it well as well when you write it down. These notes will also be handy for you for your examination or any competitive exams or any just for a revision sake. So let's begin. So this basically includes the introduction of carbohydrates and the classification of carbohydrates and a brief note on monosaccharides. These topics are really important because if you understand this in a better way, then you can understand the further topics very easily. So let's begin. So first, let's see what are the general functions of carbohydrates. All of you know that it is the abundant organic molecule which is present on the earth. And also it forms a main structural component of plants. They provide food energy in the form of starch and sugars. And also, in fact, carbohydrates are one of the major source of energy that is provided for humans through diet. And the major thing is that it acts as a metabolic intermediate, and it is a constituent of RNA and DNA. It's also a structural element of cells and tissues. And the major thing is it acts as an energy storage in a humans. The last point is really important, that is the functional diversity and the structural diversity. Because structurally these sugars or the carbohydrates have a different types of arrangement, that makes it possible for a particular sugar or a carbohydrate to perform multiple different functions in our body, especially which are related with the metabolism. Moving on to the next topic, that is what is the composition of carbohydrates? So as you all know, it is made up of carbon, oxygen, and hydrogen atoms. And they are usually referred to as hydrates of carbon. And because of the presence of functional group, whether it's an aldehyde group or a ketone group, the carbohydrates are named accordingly. So in the classification, majorly the carbohydrates are classified as simple carbohydrates and the complex carbohydrates. Simple carbohydrates includes the monosaccharide, which is a 1 sugar unit. Whereas the disaccharide has two sugar units in that. So these both are included as a simple carbohydrates. Whereas complex carbohydrates include oligosaccharides, which contains 3 to 10 saccharide units. Whereas if it contains more than 10 units, then it is termed as polysaccharides. So now you can see this flowchart, right? This is a classification of the carbohydrates, and this is the simplest form through which you can understand and you can answer most of the 50% of the questions that can come from carbohydrates. Usually in the competitive exams, they ask the simple basic questions from these classifications only. So make sure you go through this flowchart. So let's see. First is the carbohydrates is mainly divided into simple carbohydrates and complex carbohydrates, as I told you now. And again, the simple carbohydrates are classified as monosaccharide and disaccharides. So here, if you see the monosaccharides, the examples are glucose, fructose, and galactose. And these monosaccharide results in the formation of the disaccharides, oligosaccharides, and polysaccharides. So these monosaccharides cannot be further divided or it cannot be reduced or hydrolyzed. So this is the basic unit through which the other classes of carbohydrates are formed. So when we talk about disaccharide, which contain 2 sugar units, so these disaccharides can be reduced to a single unit of sugars, right? So upon hydrolysis, they give different monosaccharide units. So this is one of the major questions that can be expected in the exams. So usually they ask, what do you get from hydrolysis of sucrose? So usually sucrose is made from glucose unit and a single unit of fructose unit. So glucose plus fructose gives you sucrose. So upon hydrolysis of sucrose, we get glucose and fructose. So similarly for lactose, it is made up of glucose and galactose. So maltose is also made from two glucose units, whereas trehalose, it is also made from glucose. So here, if you can see, trihalose and the maltose both are made from 2 units of glucose, right? But why the name is different? Because the bonding of these 2 glucose units will differ. At what point is the bond formation? Based on that, these two differs. Coming to oligosaccharides. So here, if you talk about raffinose, this is formed by a combination of glucose, fructose, and galactose. So if there is a question where they ask on what hydrolysis what we get, so you can choose an option where it is given glucose, fructose, and galactose. Apart from that, if you see here, the one point which you can notice here is the dextrin, which is included in both oligosaccharides and polysaccharides. Why is it so? Because when the starch is partially hydrolyzed, we get an intermediate product called dextrin. So this is usually formed when there is a degradation of starch to maltose by the enzyme alpha amylase. So that is why it is given in between these two, polysaccharides and oligosaccharides. And here also, if you see, even starch is made up of glucose units and even glycogen is made of glucose units. So then why is the two molecules different? So again, there's a structural difference between these two. In case of starch, there is both linear and branched structure. That is, amylose is a linear structure unit of glucose, whereas amylopectin is a branched chain. Whereas in case of glycogen, the whole molecule is a branched chain. And dietary fiber is also part of polysaccharides. So if you learn this table, as I said, you can answer most of the questions. So make sure you learn this flowchart and memorize it. Going on to the next topic. So next we are looking for the monosaccharides in details. As you know, these monosaccharides are the simplest carbohydrates that cannot be further hydrolyzed even from the digestive enzymes or any chemical reactions. And they're commonly referred to as sugars, monosaccharide units, or residues. In terms of nomenclature, the suffix OSE is used. So this OSE is used in case of the nomenclature for any monosaccharide or any other carbohydrate. And one more nomenclature is based on the number of carbon atoms that is present in a sugar. So in case if the sugar is containing three carbon atom, then it is termed as trioses. Whereas if it is 4, tetroses, and so on. If it is 5, pentoses, hexoses for 6 carbon atom, heptoses for 7 carbon atoms. So apart from this, the number of carbon atoms, we can consider even the functional group that is present in the carbohydrates. So that is, if the sugar is having an aldehyde group, then it is termed as aldoses. If it is having a ketone group, then it is termed as ketoses. So depending on what basis we are naming the sugar, the names of these carbohydrates or sugar differs. So now we are looking into the chemical structure of monosaccharides. Here you can see the chemical structure of glyceraldehyde and dihydroxyacetone. So here they have given this is aldoses and here it is a ketosis. Why is it so? Just now I told you there is a nomenclature based on even the functional group. So here if you see at the carbon 1, the carboxyl group contains the aldehyde functional group, right? So that is the reason why this is named as aldoses, and here the term aldehyde is used because of the presence of this functional group. Whereas here you can see at the C2 position, there is a ketone group present. So that is why it is termed as ketosis. So these two are the smallest monosaccharides possible, and their structure is given. So let's move on to the next topic. So now we are looking into the topic stereoisomers, which is one of the important properties that a carbohydrate or sugar possesses, due to which it has a huge importance in the biological system. Because of this stereoisomer functionality, there are different functions that have been performed, that are being able to perform in our body. So before knowing what are stereoisomers, let's see what are isomers. So most of you might be knowing that. See, the isomers, they are the compounds which have the identical molecular formulas, but they have a different structure. So just remember this, isomers means they have I for identical molecular formula. Okay. IM. So here, if you don't remember that, because there are a number of different anomers and all, so you just, by the spelling, you can just see that here IM you remember. So they have identical molecular formula, but there is a different structure. Okay. So coming to stereoisomers. So they are the spatial isomers. So you know what are isomers? That is, they have a different structure but similar molecular formula, so that is same for here also, but they have mentioned a new word, that is spatial. So here you see they are the spatial isomers, meaning that two or more compounds which have a same composition, molecular composition is same, the bonds are same, but the bond differ in their 3D orientation in space. So basically the bond and the molecular composition will be same even in case of stereoisomer, but where this bond forms in the 3D structure, that will differ. Hence they are called as stereoisomers. And also this existence of this stereoisomer is due to the presence of asymmetric carbon atoms. So now what are asymmetric carbon atoms? They are the carbon atom which have 4 different atoms or groups which are covalently attached to each other with 4 bonds. So that you might be knowing in the basics, right? What is an asymmetric carbon atom? It is basically a carbon atom which has 4 different atoms or groups which are covalently attached to each other in each other of its 4 bonds. So at each side of the carbon, there will be different group. So that is said to be a asymmetric carbon atom. So in case of monosaccharide, 4 or more carbon have this multiple asymmetric carbons. So in case, if you see the structure of monosaccharides, you can find number of asymmetric carbon atoms which have a different molecules in each side of the carbon. So let's see what is next. So now we are here, we will understand the chemical structure of monosaccharides. And we'll also see which is the asymmetric carbon given. So let's take the first example of glucose. So here, as you can see, the asymmetric carbon is C2, C3, C4, and C5. Why? Because if you just see, each side of it has a different compound. So that is why we can say that C2, C3, C4, and C5 are asymmetric carbon atoms. And also, why is it this L-glucose? And why is it D-glucose? So where is the difference? Here, the presence of OH is different, right? So the chemical formula is same for both, but OH here is on the left side, whereas here it's the right side. So whenever this OH is on the left side, it is L-glucose. Whereas whenever it is on the right side, it is said to be D-glucose. And how to identify which of this, see here also there is a OH, right? Here also there's a OH and here it's OH. So here it is on the right side. So why not this as a D-glucose, right? So this doubt is that when, as per the rules, see at the carbon one, we get the functional group, right? So we need to choose a carbon which is far from this functional group. So C1 has the functional group. So here C5 is the last carbon, which is far away from this functional group. And this we have to consider for naming whether it's an L-glucose or D-glucose. So here C5 has OH bond on the left side and hence it gets the name of L-glucose. We cannot consider C2 or C3 for naming this way, but we have to consider the carbon atom which is far away from the functional group. So here also same, that is the C5. It's on the right side, so it is termed as D-glucose. So similarly, you can compare here as well. So this is the first carbon, it doesn't have a long linear chain. So here the asymmetric carbon is also C2, and this is only considered for naming whether it's L or D. So as it is, the OH group is present on the left side, it is for L-glyceraldehyde, whereas it is on the right, so it is the D-glyceride. Similarly, it holds good for the galactose and mannose. So in both the cases, OH is on the right side, and hence it is denoted with D. So let's move on to the next slide. So now we are looking to different forms of these stereoisomers. So as we have seen in the previous slide, where you can see the structure, we know how D and L form configuration are formed, how we name them, right? So if those structures exist as D and L configuration as a mirror image to each other, please do not confuse. When we say mirror images, it's not like it's superimposable. That is, it can't be kept on each other with the same structure. So if it's a mirror image and they are not superimposable, then it is termed as enantiomers. That is, they should be a mirror image to each other. For example, D and L glyceraldehyde and D and L glucose. So they are the mirror image to each other. Whereas if the stereoisomers are not even the mirror images and even they are not superimposable, then they are said to be diastereomers. So for this, the example is D-glucose and D-galactose. Let's go back to the structure and see why, how they are the mirror images and how this example, that is glucose and galactose, are not mirror images. So let's go back and see. So here, if you can see, this L and D form are there, right? So why is it mirror image? So here, it's an opposite way, right? Here, the OH group is on the left side, whereas here it's on the right side. So whenever we stand in front of the mirror, usually it's the opposite, like left hand and right hand will be opposite in the mirror image, right? So similarly, even in this structure, this acts as a mirror image. So if you keep this structure in front of the mirror, then it will show us this, right? It's same, but only the OH is different. So it is this the mirror image. Whereas here, if you see, OH is here and it's here. So similarly, everything is opposite in the L and D form. Hence, since it is possibly a mirror image, but they are not superimposable, they are termed as enantiomers. Here, if we take the example of D-glucose and D-galactose. Okay, so both of the structures here, it resembles same, right? Except HC4 carbon, right? So if it's a mirror image, then this OH group should be here on the right side, whereas this is on the left side. Hence, it cannot be representing a mirror image, and also definitely they are not superimposable, and hence they are termed as diastereomers. So now I hope you know the difference between enantiomers and diastereoisomers. That is, here it should be the mirror image plus not superimposable, whereas for diastereomers it should be not even mirror image, not superimposable. So moving on to the next topic. The ring structure. Usually when these sugars are in the solution, they don't maintain this linear form anymore, but they form this bond like a ring structure. So this is because of the reaction between the carboxyl group and the single hydroxyl group. And to be more precise, these ring structure are much more stable than these open chain. Again, similar to the open chain structure, even if this ring structure has a aldehyde group, that is like cyclized sugar, is formed with an aldehyde group, then it is called as hemiacetal, whereas hemiketal for the cyclized sugar, which is formed with a keto group. So it's a simple concept. Usually this ring structure is formed whenever the sugar is in the solution, and this is much more stable than the open structure. So as we know that in case of the open chain structure, we have asymmetric carbon atom. So similarly, in case of ring structure, there is a unique asymmetric carbon atom which arises from the carbonyl group, and it is termed as anomeric carbon. So anomeric carbon and asymmetric carbon are the same. Asymmetric carbon is the one which is present in the open chain structure, whereas anomeric is term used for used in case of ring structure. So how do we identify this anomeric carbon? So hydroxyl group, that is OH group, which is attached to this anomeric carbon, it will exist on either side of the ring structure. So which creates another type of stereoisomer, that is anomers. And it is denoted by alpha or beta. So as we in open chain, we used to denote it by D and L. For here, based on the position of this hydroxyl group in the ring structure, we name these carbon in terms of alpha and beta. So let's understand this from seeing the structure. So to understand how we give this alpha and beta name, so let's understand with this example. So here I have taken this aldohexoses, and here it's a ketohexoses. So first step is we need to identify the anomeric carbon, right? So in case of hemiacetals, that is like the one which has the aldehyde group, which is usually glucose, galactose, and ribose, the anomeric carbon is present at C1. So first carbon itself. But in case of hemiketals that contains a keto group, usually in case of fructose, the anomeric carbon is present at the C2. Okay. So coming to this now hydroxyl group. So here the hydroxyl group, it is present in the downside, right? So if it is present at the downside, then it will be named as alpha. Whereas if this OH group is above the ring structure, that is, it is indicated at upwards, then it is termed as to be beta. So it's in a simple word. So usually you have to consider C2 as the anomeric carbon to know whether OH is above or below to name alpha or beta. In case of keto group, in case of aldehyde group, C1 itself is an anomeric carbon, and if it is below, then it is alpha. If it is above, then it is beta. So this is how we can name these ring structure. Let's see what is this importance of these different structures, these stereoisomers. So basically, these stereoisomers among these monosaccharides, and even including the other nutrients like amino acid, lipids, they have an important metabolic implication. Because of this stereospecificity of certain metabolic enzymes. And also, the major important example of this is the action of the alpha amylase, which is an enzyme, which hydrolyzes the bond between the glucose units in the polysaccharide starch. So this alpha amylase is so specific to just the alpha linkages between the D-glucose, alpha D-glucose in the starch. So that is the reason why this alpha amylase can act upon these alpha linkages, which is formed in D-glucose, and starch is digested in our body. Whereas the cellulose has beta linkage. Now you understand what is alpha and beta, why they are named so, because of this structural difference. The OH group is down or up based on that. So this alpha linkage, what happens, because the specificity of this enzyme, it cannot act on this beta linkage, and hence it cannot be digested. So that is the reason why cellulose, we lack the enzyme in our body which can digest the cellulose. Whereas starch is digested because of the specificity of alpha amylase to the alpha linkage between this alpha D glucose molecules, which is present in the starch. So this is the importance of stereoisomers. This is just an example, but it has a different importance in whole metabolic activity, directly or indirectly. So let's next see about the reducing sugars. So usually the monosaccharides which are turned into these hemiacetals or hemiketals, they are called as reducing sugar, just because of their reducing capacity. Like they can reduce this copper ion like from C2 plus 2 plus to C2 plus 1, that is reduction. And this property is really helpful in identifying which end of the polysaccharide chain has the monosaccharide unit, which is like open, or it's a ring form, like it's a closed structure. Like in other words, we can say that it helps us to identify where is the anomeric carbon present. So pentoses is one of the important sugar. So the importance of this is because it is a basic material for so many of the molecules which are involved in the metabolic activities, and the important ones especially. So as compared to the hexoses, pentose sugar is provides a little dietary energy because of their availability, but they are readily synthesized in the cell from hexose precursors, and they are incorporated into the cells metabolically. For example, aldopentose ribose. It's a key nucleotide which helps in the production of these adenosine phosphates, ATP, adenosine triphosphate, ADP, adenosine diphosphate, and AMP, CAMP. And ribose, as you know, it's a constituent of nicotinamide adenine dinucleotides, that is NAD plus, NADP plus. So these ribose and its deoxygenated form, that is deoxyribose, they are the part of RNA, right? And also the DNA. So this ribitol, which is a reduction product of the ribose, and which are also involved in this vitamin riboflavin and flavin coenzymes, FAD and FMN. So these are very important. So pentose holds a very crucial part of many of the metabolic activities. So let me know if you know these abbreviation, that is ATP, ADP, AMP, CAMP, NAD plus, NADP plus. in the comment section. So moving on to the next slide. So now we learn about the derivatives of monosaccharides. So monosaccharides in the cell itself will modify itself in order to perform a specific function. For example, just now I told that this ribose will be modified by just reduction or deoxygenation, which will form this deoxyribose or ribitol, which performs a specific function. And for example, these modification of the monosaccharides in the cell is really essential to form the larger molecules, and even it combines with other nutrients like, for example, proteins. Protein plus sugar will be formed for many of the metabolic activities. So one such example is proteoglycans, which is a addition of protein plus the sugar, and it will which contains this glucuronic acid as well as acetyl galactosamine. So these together are found majorly in the connective tissues, which includes even bone and cartilage. And including there are dietary supplements that are provided in order to increase the strength of joints. So this is used for the marketing purpose. So joints, these dietary supplement which contains this galactosamine are marketed to support the joint health. But despite there is a little evidence to prove that these galactosamine will be effective for this bone's health, but these two, this is the protein plus sugar content, which is modified from the monosaccharides in the cell, are abundantly found in the connective tissues. So this was all about the introduction for carbohydrates, classification, about the structure, that is stereoisomers, ring structure, and derivatives of monosaccharides. So in the upcoming video, I'll be covering the disaccharide and syrups, which is the liquid sugar. So if you like the video and if you understood the concept, then do subscribe and hit the bell icon so that you can get the notification whenever there is a new video uploaded on this channel. You can also follow Food Feeds in many other platforms like Instagram, Telegram, where there's a constant update on the food technology and aligned field. So hope to see you in the next video. Thank you.